Cocaine and its derivative, crack, amphetamines, and antidepressants are all drugs that provide energy, confidence, and a sense of euphoria. They are known as psychostimulants. In the mountains of South America, the coca plant has been venerated as sacred for thousands of years. Under the reign of the Incas, it was burned and consumed during ceremonies in honor of the Sun King. It is still used in divinatory rituals by Indian shamans even today. The indigenous peoples of Colombia, Bolivia, and Peru have traditionally chewed the leaves of the coca plant to give themselves energy, resist fatigue, and ward off hunger. Rural families can legally grow coca for their own consumption or for use in medicines. However, by selling their harvests to clandestine laboratories that manufacture cocaine, they can more than double their income. It is estimated that there are over 13 million cocaine addicts in the world today. With 158,000 hectares given over to illicit cultivation, cocaine has become a huge business, generating billions of dollars in profits and subjecting entire countries to the domination of drug traffickers.
Esta es la hoja de coca. Este de aquí es su droga, lo que sale. Esto de acá. Esto una vez que se le pisa ya comienza a salir, mira. Sale la droga. Su nombre es el... Primero le ponemos esto, o más.
But before it can affect the brain's delicate chemistry, some crude chemistry must be performed, involving large doses of lime, sulfuric acid, and kerosene. A very potent derivative is then made from cocaine, called crack, which is sold in the form of small rocks.
En vivo y en directo para que prendas pues estas notas, esto no se toma. Eso se llega hasta nuestro paladar, hasta aquí nomás y se le bota. Ahí va, ahí sale la...
¿Has probado el palo? ¿Sí? Sin topar la cuchara tengo que... Ya lo ve.
Go ahead, from your side, Gene, feedback.
Not far from New York, the Brookhaven National Laboratory is one of the only places in the world where researchers like Jean-Jacques Wang can study the real-time effects of drugs on the human brain.
We know that cocaine being used for thousands of years by South American Indian. They use a chewing tobacco. It really is very enforcing. It's not considered as an addictive drug. But once they become crack and they snorting, because of the speed is not so fast, it's not so reinforcing. But smoking and the IV is very, very reinforcing. So you can know that the route of administration and the speed of the cocaine delivery into the brain is one really make the person become addicted. Usually the people in the New York area, they use a pipe with a filter. And the crack, once they purchase the crack, they put a small amount of the crack in, just in the tip. And then they use a lighter and then they inhale, and then keep a breath, and they exhale. And the speed of the crack will go through the mouth and into the lung, and then into the brain immediately.
The scientists at Brookhaven often use positron emission tomography, an imaging technique that slices up the brain virtually to identify the areas activated by drugs. Here it shows that crack invades certain areas in the brain at a much faster rate than injected or powdered cocaine, which explains the formidable intensity of its effects. These studies were done by Nora Volkow, a researcher who oversees all government-funded research on drugs in the United States.
We are showing with imaging technologies, and certainly other investigators as well, using imaging but also in studies in animals, that drugs, repeated administration of drugs, disrupts the way that the brain works in very fundamental ways that affect not only our motivation, the motivation that the person has to take the drug, it makes it extremely intense, but it also affects areas of the brain that are involved with operations such as making decisions, judgment, our ability to control our acts. It also affects memory circuits. And not just the memory circuits that we use to learn a new language, but the memory circuits that are involved in recalling emotional experiences.
Inside the cranium, the most visible part of the brain is the cortex, considered the seat of intelligence. Buried under its folds is the limbic brain, which governs our emotions and motivations. This is where drugs go to work. All drugs work by hijacking the brain's communication system. Our brain, indeed, spends most of its time chatting to itself. In order to keep things running, it relies on a network of ongoing conversations between our 100 billion nerve cells, or neurons. The neurons exchange information by sending each other chemical messengers called neurotransmitters. Whether it has to do with pain, pleasure, or the rate of information exchange, the neurotransmitters act as regulators, maintaining a complex and fine-tuned equilibrium which is essential to all of our bodily functions. Between the neurons are little spaces which are always busy with activity. It is here, in the synapse, that the neurotransmitters do their work. They transfer information from one neuron to the next by binding themselves to the appropriate receptors. These systems exist in human beings and in all mammals, including the smallest of them all. Studying how drugs work would not have been possible without rats or mice. They share many of the same genes as human beings, and drugs stir up the same kinds of tempests in their brains. In fact, all of humanity is morally indebted to these little creatures. We can intoxicate rats, manipulate their genes, even cut them up into little pieces. This is the terrible but necessary task of laboratories that compare the reactions of normal or control rats to so-called knockout rats. This technique consists of knocking out certain genes in normal mice, for example, the gene that controls the production of a certain neurotransmitter. These genetically modified mice are called knockout mice. Dozens of neurotransmitters have now been identified, but the one that plays a key role in feelings of pleasure and the desire for drugs is the aptly named dopamine. This is what dopamine looks like under a microscope, in its crystal form, and its chemical structure. Bruno Giraud studies the wide-ranging effects of this tiny molecule in the brains of normal and knockout mice.
Toutes les drogues qui sont All drugs taken by human beings, nicotine, alcohol, opiates, cannabinoids, psychostimulants, have one thing in common, which is that they stimulate the release of dopamine. So dopamine is at the core of the brain's reward circuits, as well as, of course, its pleasure circuits. Dopamine is also released by compulsive gaming, sexual activity, and eating, for example. In fact, sexual activity and eating are very important for the production of dopamine. In other words, they are survival behaviors, necessary for the survival of the species. What drugs do is imitate these activities. They hijack the system, they take advantage of it. The brain wasn't originally made to be dependent on drugs. It was made to manage the reinforcing systems that lead us to engage in sex and eating. Drugs work by deceiving and taking over the system.
This experiment reveals one of the functions of dopamine. The box on the right contains a normal mouse. On the left, a knockout mouse, which lacks a key element in its dopamine circuit, the transporter. Unlike the normal mouse, the knockout mouse seems ready to jump into the ring. If cocaine is injected into the normal mouse, it starts to behave in exactly the same way.
Here you have the nerve ending of a normal animal, which releases dopamine. The dopamine will then bind to receptors on the receptor cell here. And quite quickly, the dopamine is recaptured by the dopamine transporter and taken back into the neuron. In the normal mouse that has been injected with cocaine, the cocaine, which, like amphetamines, is a psychostimulant, will block the dopamine transporter. In the knockout animal, you have everything. The nerve ending, dopamine release, activation of receptors, except the reuptake. So a lot of dopamine remains here in the synapse, and it overstimulates the receptors, which is why you see this hyperactivity. These mice are much more active. Their behavior is in fact identical to that of the normal mouse you saw who was injected with cocaine. Once we've understood how the system works, we can identify which levers to push in order to increase or decrease the amount of dopamine that is released. Afterwards, we can test our hypotheses on human subjects using the pharmaceuticals that we have or those that will be developed in the next few years. Obviously, with this animal model, we can't ask the mice about their mental disturbances, which is what we want to explore in human beings. But everything that's behind these mental disturbances, the neurobiology, is exactly the same. It's just that the neurobiology is expressed differently. In mice, for example, through movement, and in human beings, through mental confusion.
By observing how these mechanisms work in control and knockout mice, researchers have identified the many different functions of dopamine in the brain. And when they slice up their minuscule animal brains, they are taking a first step towards discovering new medications.